Hello, Cell and Genetic Biology 230. This is Dr. Chastity Bradford, and we are covering cell communication, chapter 11, and this is concept 11.1. Many of us communicate in a number of ways: Instagram, through our cell phones, texting. Our communication with people where we text is sometimes can be local communication. Someone could be right next to us and we could send a text message. Someone could be across the globe and we could send a text message. We often communicate using Twitter. And so we have evolved to become people who communicate in a number of ways. And what I want to do today is to provide an overview of cellular messaging and to discuss how external signals are converted into responses within cells. So cell-to-cell communication is crucial for not only multicellular organisms but also unicellular organisms. And what's interesting is that scientists have discovered that there are some universal or some common mechanisms of cellular regulation. Cells most often communicate with each other by chemical signals. For example, the fight or flight response is triggered by a signaling molecule called epinephrine. So imagine how this animal feels when he is being chased by this predator. There's a lot of signaling that's going on in his body. Epinephrine, sometimes called adrenaline, will bind to its receptor and it will signal, transduce a signal throughout the cell. And the response will be the production of glucose from glycogen in the muscle tissues of this animal so that it can move as fast as necessary. So signal transduction pathways, they convert signals on the cell surface into cellular responses. This is an example of how Adderall, a common medication used to treat hyperactive disorder, ADHD, it's often what we call it. Adderall is a mix of two stereoisomers of amphetamine. So the term stereoisomers should be really familiar to you. The chemical structure of Adderall is close to epinephrine, and I just showed you in the previous image of how epinephrine works in the fight or flight response. The response as a result of Adderall is alertness, euphoria, oftentimes insomnia. But as with any medication, there can be side effects, and the side effects of Adderall are anxiety and psychosis. And one of the things you'll notice in the magazine that you receive from the bookstore, every time you purchase something, there is a little magazine inside. And one of the things that I noticed recently was a lot of ads in there for adult attention deficit disorder, hyperactive disorder. And so I want you to just peruse it and see if you find that there are a number of ads in that magazine. And so what we have here are two neurons and a synapse, which is just the space in between these two neurons. And depicted here is a transporter, not important for this conversation. Adderall is shown in as a red square, catecholamine shown as the C's here, and then the receptor for catecholamine shown as a Y, a red Y. And so what is happening is normally there will be catecholamine released into the synapse, and catecholamine will bind to a postsynaptic neuron, and then a signal will be transduced and a certain response will occur. And because Adderall is similar to epinephrine, similar to the catecholamines, the drug itself will also bind to catecholamine receptor to produce the same response or similar response. And so a lot of drugs are designed to mimic signals that are normal for our body. And our body has a way of converting these signals to responses within the cell. Yeast, specifically the yeast that we find in bread and wine and beer, is noted here. And this yeast has two different mating types. There's an A and there's an alpha mating type. And as you notice here on this yeast cell, A, there are certain receptors depicted here in brown. And on this other cell, the alpha cell, there are also receptors there too. And so what this illustrates is how cells communicate, and even in mating, cells have to communicate. And there is a signal, there is a transduction of a response, and then there is some type of a transduction of a signal, and then there is some type of response. Okay, and so what is happening here is these yeast are mating. So in order to mate, they have to locally secrete signals or factors, if you will, ligands, if you will, to each type, each mating type. And when that occurs, the yeast mate, as you see here, and produce a new yeast, a new cell. And this new cell combines the material from the A and the alpha yeast cell. And so now you have a new A alpha cell. And this occurs because of signal transduction pathway. And what is a signal transduction pathway? It's a series of steps by which a signal on the cell surface, as you see here, this ligand binds to this receptor. This ligand binds to this receptor. So the ligand, or the signal, if you will, from the yeast cell mating type A will bind to the receptor on the yeast cell mating type alpha. And the alpha factor, or the ligand that is released from the alpha cell, then binds to its receptor on the A cell, and then mating takes place. And every time we have a ligand binding to a receptor, there is a high degree of specificity there at this site, this active site where the ligand binds. Okay, this is one level of control for the cell in terms of communication, that only this this A factor will bind to this receptor, and only this alpha factor here will bind to the receptor. So receptor ligand communication is very specific. And so a signal transduction pathway is a series of steps by which a signal on a cell surface is converted into a specific cellular response. And the response in this instance, of course, is mating. Cells in multicellular organisms communicate by chemical messengers. Animals and plant cells have cell junctions, and this is something that we talked about previously when we identified the various organelles within animal and plant cells. Now, these junctions can directly connect the cytoplasm of adjacent cells. And in local signaling, animal cells may communicate by direct contact. They may communicate by cell-to-cell recognition, or there may be chemical signals released. What we find here is an example of local signaling. These are two different cells. Of course, this would be your animal cell here on your left, and what we have here on the right would be a plant cell, noted by, you can kind of tell that, of course, there's some plant wall and there's some green, probably chloroplast depicted here. Okay, so you have your plant and your animal cell. And one of the common things that you notice here is that they can communicate via direct contact. Do you recall how what junction occurs in animal cells that allows their cytoplasm to communicate, or things to be released from this one cell's cytoplasm to the other and vice versa? Do you recall what junctions exist, cell junctions exist in plant cells that allow their cytoplasm to communicate? Well, I'll help you out. Gap junctions between animal cells allow their cytoplasm to communicate, and plasmodesmata in plant cells allow the plant cells, adjacent plant cells, to communicate. These are cell junctions, and they're responsible for that local signaling via direct contact. Now, there's also other local signaling that can occur via direct contact, and this can occur in response to the presence of certain glycolipids or glycoproteins on the cell surface. And if you recall, you should recall why these glycoproteins are on the cell surface. They allow one cell to recognize the other cell. And so these are two methods of local signaling as it relates to direct contact. There's cell junctions and also cell-to-cell recognition, which involves glycolipids and often glycoproteins. Now, there are three types of chemical signaling. This is very important. You will need this information for the rest of your life, lifelong learning. One of them is autocrine signaling. In autocrine signaling, the cell signals itself, auto-self. It signals itself through a chemical that it synthesizes and then responds to. Autocrine signaling can occur solely within the cytoplasm of the cell or by a secreted chemical interacting with receptors on the surface of the same cell. Cell signals itself. There's also paracrine signaling. In this case, chemical signals diffuse into the area and interact with receptors on nearby cells. Autocrine signaling is self-signaling. Paracrine signaling is local signaling within an area, a distinct area. Okay, so examples are the release of cytokines. They can cause an inflammatory response in a local area. The release of neurotransmitter at synapses in the nervous system, as depicted with the Adderall example, is an example of paracrine signaling. Now, there's also another type of signaling. It's more long-distance signaling. This is endocrine signaling. This is the type of signaling where chemicals are secreted into the blood, carried by the blood and the tissue fluids to cells that they act upon. So that is long-distance signaling. Okay? Local signaling. So, as I mentioned earlier, sometimes people are sitting right next to each other and texting. That's local signaling. Are you sitting right next to someone and you're communicating to them? That's local communication. So communicating cells, they can be close together or these cells can be far apart. Remember, we're talking about multicellular organisms and they have to release signaling molecules to target other cells. Okay? So what we're seeing here is local signaling, and you have a cell here and you have three other cells, and these three other cells are going to be called target cells, and this cell will be the secreting cell, if you will. And inside the secreting cell there's a secretory vesicle, and this is something that we discussed when we talked about organelles. And local regulators, so that with local regulation, the secretory vesicle releases, as you see here, a signal, okay? And then this signal then binds to its receptor, somehow communicating with this target cell that's nearby. So this is paracrine signaling, where there's a local regulator that diffuses through the extracellular fluid, okay, to communicate to a nearby cell. This is another example of paracrine signaling, another way of seeing it. You have a signaling cell and you have a target cell. That target cell has to have a receptor. And so the signaling cell secretes or releases its signaling molecule or a ligand. That ligand binds to the receptor on the target cell, a local target cell. Autocrine signaling, on the other hand, is when a cell secretes or releases a signaling molecule or a ligand, and this signaling molecule then binds to a receptor on the cell, on the same cell that released it. There's another type of local signaling, and that's synaptic signaling. And that involves in this instance, a nerve cell, a synapse, which is just a space in between two neurons, two cells, neurons or nerve cells that are communicating with one another, and of course the other neuron or the target cell. So there is an electrical signal that comes along the nerve cell, and it triggers the release of a neurotransmitter. This ligand is the neurotransmitter. It diffuses across the synapse. It diffuses across this space. And remember, we talked about diffusion. And so it diffuses across the synapse until it reaches the target cell, and it stimulates that target cell. So these are three mechanisms of local signaling. There's paracrine, autocrine, and synaptic signaling. So in this instance, what's the chemical signal? In this example, what will the chemical signal be? It would be the neurotransmitter. In this instance, what would be the chemical signal? It would be the molecule that's secreted, right? Now, in addition to this local regulation, this local signaling that occurs when these messenger molecules travel just short distances, and by the way, there are often growth factors are often the signaling molecules that are associated with local signaling. Now, in addition to this local signaling or local regulation, there's also long distance signaling. And this takes place in both plants and animals. And the chemicals that are used for long distance signaling are called hormones. Now, with long distance sometimes you may hear it called endocrine signaling, more often than not. Endocrine signaling, the chemicals are secreted into the blood. They're carried by the blood and tissue fluids to the cells that they act upon. So in this instance, this is an endocrine cell. It releases its chemical into the bloodstream. This is a blood vessel in pink. Now this chemical that's released, or hormone, it travels in the bloodstream, okay? And then it travels until it binds to the cell that it's supposed to act upon, okay? And then, of course, you have your response. So again, you have a signal that is transduced, and then you have some response. And when this takes place in a manner that requires the secretion into the blood and traveling for a long distance to the responding cell, this is called long distance or endocrine signaling. So in this instance, what's the chemical signal? The chemical signal, of course, is going to be the hormone. Now, there are three stages of cell signaling, and I have alluded to those throughout the beginning of this lecture. And these three stages were discovered by Earl Sutherland. At least he was given credit for discovering how epinephrine acts on cells. And he suggested that cells receiving signals went through three processes: reception, transduction, and response. Reception, transduction, and response. Now, what are receptors and what's reception? What's that first step? Reception. In reception, a chemical signal binds to a cellular protein, typically at the cell surface, or it could happen inside the cell. So this signaling molecule, which we call a ligand, binds to its receptor. And in this instance, this receptor is in the plasma membrane on the outside. So this ligand is binding on the outside of this cell. Now, this signaling molecule can also move and travel through the plasma membrane depending on its size and depending on its chemical makeup and structure. And so this is not always how it happens. So it can happen intracellularly as well. Okay, but the first step in the process is reception. Okay, the second part of the process is transduction. The second component of signal transduction is transduction. Okay, and in transduction, it is binding. In transduction, binding leads to a change in the receptor. So this receptor often changes its shape, or conformation is something, another term you'll hear us say often. And that triggers a series of changes in a series of different molecules along what we call a signal transduction pathway. Signal transduction pathway. Now, these molecules in this pathway are called relay molecules. Okay, so we have reception, we have transduction, and the third step is a response. Okay, and that response can be a number of things. It could be transcription of a gene. It could be growth. Okay, and it could be apoptosis, which is programmed cell death. It could be proliferation, which often happens in cancer cells. And so that is the third step in the process. There's reception, there's transduction, and there is response. And all three of these you must know. Okay? Because in the response, of course, the transduced signal triggers a specific cellular activity. So now, we have a neuron, and it responds to a particular neurotransmitter by opening gated ion channels. The neurotransmitter is serving as what part of the signal pathway? Now, we've already explained what a receptor is. We've explained the relay molecules in the last slide. Transduction or transducer is part of that relay. And then you have the signaling molecule, the ligand. So what is a neurotransmitter serving as?